Our next speaker this afternoon is Dr. Daniel Gale. Danny is an associate professor and honorary consultant nephrologist at the Royal Free Hospital. He's going to explain to us how genetics can impact on IgA nephropathy.
So thank you for the opportunity to talk to you, and thanks for listening to me today. So I'm really going to give you some background about genetics, what we mean by genetics, and how that relates to IgA nephropathy. So genetics is about genes, and genes are the unit of instruction that our cells carry in order to make the proteins that are the building blocks for cells that give them the ability to do all the things that our cells need in order to come together and form a functioning organism like a person. Each cell in our body has got 2 copies of almost all of our genes, but only a proportion of those genes, those instructions, are switched on at any one time. And it's the particular genes that are switched on that determines which proteins are being made by a cell, and that determines whether the cell is making fingernail or in your eye or in your ear or in your brain or behaving the way it should. But the important thing is that all of our genes are present in almost all of our cells all the time. Genes are encoded in a chemical called DNA, the structure of which was discovered in the 1950s. And DNA is quite a special substance because it forms this rather beautiful double helix, which you may have heard of, and it comes in 4 different letters or bases. So these are different DNA molecules that stick together into a double helix, and they are made by bases called adenine, thymine, cytosine, and guanine, or A, T, C, and G. And it's the sequence of A's, T's, C's, and G's in this molecule that is the genetic code that encodes all the genes. And it's just like a book, in that each chapter is a gene. The chapter is made of a series of letters that come together to form a word, a sentence, and a chapter of the book. And these are genes joined together in very long molecules called chromosomes. And the observant among you will have seen that A's always pair with T's, C's with G's, and so this code, you have 2 strands of DNA that stick together, and that allows the cell to then replicate the DNA, and that's the sort of fundamental building block for life. So this is another picture that shows the DNA molecule with the sequence of, we call them bases or individual letters, and it's wound up really tightly on special proteins, and they all come together to form chromosomes, and we have 23 pairs of chromosomes in almost all of our cells. If you unwrapped the chromosomes in a single cell and put them, all the chromosomes end to end, you'd get 2 meters of DNA. So that's how tightly it's lined up. And because we've got around 35 trillion cells in our body, that means that if you put all the DNA from all of our cells together, it would stretch twice across the diameter of the solar system, which is too big a number for me to really imagine. But just to give you some idea of how tightly packed up this molecule is. Now, the genes are arranged, the name for the collection of genes in an individual is called the genome, for fairly obvious reasons. And the genome, which is the book of life, and there's a printed out version of the human genome upstairs in the next door building in the Wellcome Trust. You can actually see it from the street. And it contains 3 billion DNA bases. So we've got 2 copies of our 3 billion letter encyclopedia that is the instruction manual to make a human being. The way they're arranged, genes actually only cover a small proportion of the DNA sequence. There's big gaps between each genes, and those gaps probably play an important role in which genes are switched on at any one time. And the genes themselves have small parts that are eventually made in some substance called RNA and then copied into protein, and that's how the cells make the protein. The details are not really important, but what is important is that the non-coding regions of our genome occupy 98%, and the small chapters that actually tell us how to make the proteins that build us are only around 2%, and only a small proportion of them are known to be associated with any disease. So that's the basic structure of the genome and the genetic material that we all have in our cells. The genomes of any two people, apart from identical twins, are very similar to each other. Identical twins are almost 100% identical for obvious reasons, but for the rest of us, any pair will have between 99.6 and 99.9% identical sequences. And this differences, this very small proportion of difference, explains why we all look, sound, smell, act, behave slightly differently. It accounts for some of that variation. There's, of course, how you're brought up will affect other things, affect how you behave as well. The human genetic sequence is also 99% identical to that of a chimpanzee. So the amount of, most of the DNA that we've got in our bodies that makes proteins is shared with animals that are closely related to us, and the further across the tree of life you go, the more dissimilar animals are. But clearly most of the things that a chimpanzee has to do in order to stay alive is very similar to the things that we have to do to stay alive, and there are small differences there, obviously critically important in terms of how the animals look and behave and all the things that make us different from chimpanzees, but it's only encoded by a very small proportion of difference in our genetic sequence. So that 0.1 to 0.4% difference is at around 4 or 5 million particular places in the genome where letters tend to be different between individuals. And about 3 million of these are common variants, and we call these single nucleotide polymorphisms, or SNPs. And I'm going to use the word SNP a little bit later on in this talk, and I'm not talking about a political organisation north of the border. What I'm talking about is the normal variation between individuals. And SNPs, there are other sorts of variation as well, but SNPs are the most important ones, or the most numerous ones, and about up to 3% of them actually have a function. So, for instance, the difference in eye colour that you'll see around the room is because of SNPs, so polymorphisms, single nucleotide polymorphisms, or SNPs that don't have are not dangerous in themselves, but just account for the differences between people. Many of the SNPs don't encode any difference at all. They're just differences in the letter, rather like a misprint in a book that doesn't change the sense of a word is not going to change the understanding of the reader. So that's the genome and the genetic material, how genomes differ from each other, and how does this manifest as in sort of everyday life? Well, most of us are quite familiar with the idea of genetic traits or genetic diseases, which is where a characteristic in one member of the family is then passed on through other members of the family. We sometimes call these monogenic traits or Mendelian traits after Gregor Mendel, who first described them in pea plants in the 19th century. So one copy of our genome, you'll remember that I mentioned we've got 2 copies of our genome in all of our cells. We pass 1 copy of our genome to each of our children, and the genome is shuffled up in that process, so it's a different half of our genome to each child that we have, and they get the other half from the other parent. So they have half of their genetic material in common with each parent, but which half it is not known before is not predictable. Some genetic variants, such as SNPs, can have a big effect on the function of a gene, and eye colour is a really good example of this because it's something that doesn't cause disease, but is very obvious. And there are 2 common variants. There are less common variants as well, but there's 2 common variants encoding brown and blue eyes. And if you have either one or two brown versions, and versions of variants we call alleles, but that just means version of a gene. You have brown eyes. If you've got two blue versions, you've got blue eyes. And that means that if two people who have one brown and one blue-eyed version of the particular gene come together and have children, they will each have brown eyes because they've got one brown version. That we call as dominant over the blue version. And the children might inherit two brown copies or one brown copy from one parent and a blue copy from the other, or vice versa, and they will all have brown eyes. But the fourth child, and of course it's random, you might have 10 children and none of them inherit both blue-eyed versions, might have blue eyes. And that's sort of the simplest model of genetic inheritance of single gene defects, and that's what we mean when we talk about genetic diseases. And I've put that there because not everyone is clear about where the difference between a genetic disease is and where a disease that has a genetic contribution is. And I'm going to move on from single gene defects and talk about what genetics call complex trait genetics. And all this means is these are traits in which genes make a contribution, or genetic makeup makes a contribution, but it's not as simple as one gene, one disease, or one gene, one trait. It's the constellation of gene versions that you inherit that determines, say, your risk of disease. And we know from talking to patients and probably talking to each other that some diseases like heart disease, cancer, dementia do tend to occur in families. They tend to, people will either have lots of people in their families being affected by these conditions or maybe not so many. And clearly there's a genetic contribution to it. But these diseases, by and large, are not directly inherited from a parent such that children have a high likelihood of getting the disease. It's just increased compared to the general population. And the biological process that's underlying this is the inheritance of multiple copies of multiple different genes that each carry a slightly different risk of the disease with them. And one example of such a trait is height. So we know that height is inherited because, in general, tall people have tall children and small people have smaller children. But if you look carefully, you'll see that very tall parents will tend to have tall children, but they won't tend to have very tall children on average. Likewise, professional athletes will tend to have sportingly talented children, but they won't tend to have professional athlete children. And the reason for that is to do with the multiple different genes that encode for each or that contribute to these sorts of traits. So taking the example of height, what I've got here is each of these circles represents a gene. It doesn't matter what the gene is. And the green copies are the ones that just boost your height by a very small amount, say an extra 2 centimeters. And if you the more of these green ones you have, the more you're genetically programmed to have a greater height. And you'll see that some people have 2, some people have 3, some people have 1, and there will be slight differences in their height. And every so often a person will come along who's got 6 of these very tall genes, and they will be quite a lot taller than the general population. Now, when each of these people, so let's say these two individuals, a man and a woman, say, have a child, they pass on half of these to each child, and this child, for instance, will inherit just 1, but they could equally have passed on the other set of genes that they've got, in which case the child might have inherited 2. And likewise, in this pairing, they will, this child, say, will inherit 3 alleles, so will be similar to this parent. But if this parent, who's got 6 alleles, passes on 3, it doesn't matter who they reproduce with, there's no way the child can get, or it's unlikely for the child to get a similarly large amount. And this is because this causes what we term regression towards the mean, which is that the risk in an individual, the genetic risk in an individual might be high, but with each child, it tends to get closer to the general population's risk. And that, I think, is important for height, but also important for diseases like IgA nephropathy, where it's not the case that you inherit you either do or don't inherit a gene that causes the disease. It's that you're born with a set of genes, some of which encode a somewhat increased risk. And scientists and clinicians have been looking a long time for single genes that cause IgA nephropathy, and none has been found. That doesn't mean that there definitely aren't any, but science is not aware of any single gene changes that directly cause IgA nephropathy. But what they have seen is that IgA nephropathy does crop up in families more often than you'd expect by chance. And there's very clear differences in the prevalence, the rate of IgA nephropathy in different countries. So in East Asia, for instance, the rates of IgA nephropathy are quite considerably higher than in Europe and America. And this risk also applies to people with East Asian ancestry who live in the UK. And that very clearly suggests that there's a genetic contribution to this. But it's not like eye colour. It's not something that you pass on directly to your children. It's a risk that in future generations tends to approximate the general population's risk. So why study the genetics of IgA nephropathy? The advantage of studying it is that it gives you some insight into what the underlying biology of the disease is. It's not a way of testing people or directly offering them a cure, because it's not telling a simple explanation of what causes the disease. Rather, it's a way of seeing which genes are important, therefore, which are the biological processes associated with those genes that are contributing to the disease. And in order to do these sorts of studies, you need lots of people. Lots of people with IgA nephropathy, for instance, and lots of people without IgA nephropathy. And then with modern genetic technology, one can sample a large number of different genes and see which alleles are of a higher frequency in people with the disease compared with without the disease. And the findings from this study, which I'll show you, these sorts of studies, which I'll show you in a minute, tell us that each particular variant contributes a small amount of risk. You know, we're talking 5% to 20% increased risk, depending on if you summate all the different genetic variants that people have. And very clearly, we know that people with a high genetic risk may, for reasons of their environmental exposure or reasons we don't understand, never actually get the disease. And that some people who have a low genetic risk may still go on to get the disease. And that's just the fact that we're dealing with risk factors, we're not dealing with direct cause and effect. And this means that this sort of genetic information is really not terribly useful in an individual person. If you take a person with IgA nephropathy and find out whether they were at high or low genetic risk of the disease, it may be interesting for them on an academic level, but it doesn't take you forward in terms of making clinical decisions, and it doesn't really inform what the risk to their family is, which is slightly counterintuitive. But it is very useful if you want to understand the disease. So this is the result of the first genome-wide study that was done in the UK, published almost 10 years ago now, and this shows association at one place in the genome. So this graph is showing you every chromosome, and it's sampling about 300,000 different variants across the whole genome. So it's a very small sample of the 3 billion letters that we've got. But what it shows is that certain genes at this one place in the genome are associated with the disease. And that place is in the what we call the HLA locus, which is the part of the genome that one matches when one does a kidney transplant. So this is part of the immune system that tells people, that tells the body which proteins to attack and which not to attack. Subsequent larger studies have been done in China and the United States, and they've replicated this original finding and also identified other genes. The gene names are not terribly important at the moment, but what it's showing you is very clearly there are certain HLA types or tissue types that are associated with IgA nephropathy, and this probably provides at least some of the explanation for the increased recurrence risk in very closely matched kidney transplants. Subsequently, these genetic studies have all been put together to give a sort of catalogue of the genes, variants of which seem to confer a significant increased risk of IgA nephropathy. And if you look carefully at all of those different genes, almost all of them are known to be involved in the immune system. Some of them are involved in how our bodies make antibodies, but other of them are involved in how our blood kills microorganisms. Others of them are involved in how our surfaces defend themselves against infection. So it seems, just from looking at what the genes are, one can see that the genes that are important, that are different in people with IgA nephropathy, are genes that are important in fighting infection. And you can compare those genes with genetic associations in other diseases. Again, the details aren't important, but there's overlap between IgA nephropathy and some other autoimmune type diseases. Again, diseases where the body's immune system is slightly overactive in one way or another and causes damage to the body. And one can also look at the burden of these risk alleles, or the versions of the genes that are associated with disease, and see that they correlate really quite strongly with the diversity of infectious worms in an environment. So we're talking about pinworms, hookworms, that are not terribly common certainly in modern Britain, and weren't that common in pre-modern Britain, but were a lot more common, and importantly, a lot more different types of infectious worms were and are present in parts of the world where IgA nephropathy is common. This doesn't prove it, but it does suggest that our body's ability to respond to and destroy certain types of infections is what has driven the predisposition towards getting IgA nephropathy. So it's an upregulation of our immune system, and it seems to be associated or correlated with the types of infectious organisms there are. We can also use the genetic information in another way and make a genetic risk score. Now, I said before that adding up the genetic risk in an individual isn't terribly helpful. But if you do it in a cohort of people, then it can provide some important insights. I should say that there are so many risk alleles, versions of high risk that have been described, that almost all of us will have one or more of these risk alleles, whether or not we've got IgA nephropathy. But what we can do is compare the distribution of these risk alleles in different groups of people. And one way that this has been done is looking at the frequency of these risk alleles across the world. And this fits with the infectious disease hypothesis, in that the parts of the world where there's a great diversity of infectious parasites have a higher risk score for IgA nephropathy, and there's a lower risk in other parts of the world. It's not quite as simple as that, because the risk for IgA nephropathy is much lower in people of African ancestry, and there are likely to be good reasons for this, not all of which are understood. But we can also look at the genetic risk of IgA nephropathy in the general population. And there's a national study running at the moment called the UK Biobank, which recruited around 400,000 people from the general population, so they were unselected, and tested their genes looking for common variants. And we can look in that data and find what every individual in that study's genetic risk score for IgA nephropathy is. And what we can see is that the people in the study, the 400,000 who don't have IgA nephropathy, that's the distribution of their risk score. So it hovers sort of around the 3.9 level, and that's just an arbitrary number that's made by adding up the number of risk alleles in each person. And people with IgA nephropathy, the average risk score is somewhere between 4 and 5. So you can see that if you, as an individual, have a risk score of, say, 4.1, well, you could equally be from the general population or from the population of people with IgA nephropathy. It's not terribly helpful. But if you look at tens or even hundreds of thousands of people, you can see that these populations are different. And we can do that to look at people in UK Biobank. So 13,000 of them were known to have hematuria, or blood in the urine, which we know is one of the features of IgA nephropathy. There are other things that cause blood in the urine, and these people had only blood in the urine, and they'd never been undergone a kidney biopsy. So the cause of their blood in the urine wasn't known. And what you can see is that there's a very slight, so that's the healthy population without blood in the urine or kidney disease. So 320,000 people have a genetic risk score of 3.9. people with blood in the urine have a very slightly increased genetic risk for IgA nephropathy. And this genetic risk goes up if you've got protein in the urine and if you've got high blood pressure as well. And if you've got a diagnosis of IgA nephropathy, which is a much smaller number, then the genetic risk score is very much higher. And what this is telling us is that, well, one explanation for this observation is that 20% of the people in UK Biobank with blood in the urine, their increased genetic risk would be explained by them having IgA nephropathy that's not been diagnosed. And the implication of that is that IgA nephropathy is a lot more common than we generally recognise, and it's also a lot more benign in that there's this quite large, 20% of this 13,000 people out of a sample of 400,000, quite a large number of people who have blood in the urine, probably have IgA nephropathy. We don't know because we can't biopsy them, but have the genetic risk for IgA nephropathy, and that would be explained by them having mild IgA nephropathy that presumably will never do them any harm. So that's a whistle-stop tour of some of the things that genetics have taught us about IgA nephropathy, and I'm very happy to take any questions.
Just on your last slide, just clarify the number in the sample, the overall sample.
Yeah, so this was about 400,000 people in UK Biobank. 320,000 didn't have any evidence of kidney disease. 13,000 have just a little bit of blood in the urine. So it's quite common. About 1% of the general population are thought to have that as well.
What's the hypothesis with regards to the abnormal pattern of glycosylation on IgA and how that works in genetic?
So I didn't have time to talk about that, but we've done some work in the last few years that shows that there is a very clear genetic driver for IgA glycosylation. In other words, there's variation at certainly one gene and probably a second gene that controls the sticking on of that glycosylation group onto the IgA. And differences, the genetic difference between people in one particular gene seems to explain the very strong heritability, which is the inheritance of abnormal IgA glycosylation. That signal doesn't come through as a genetic risk factor for IgA nephropathy, and that's because the studies that I showed you before had up to 3,000 or 4,000 people in them, and you'd need studies of 20,000 people in order to test the hypothesis directly that genetic predisposition to galactose-deficient IgA is a risk factor for the disease. And that's work that's going on at the moment and might be able to give us one day give us a really good answer to your question, which is what is the causal role for galactose-deficient IgA in the disease?
Hello, this is something that you have been speaking about in the talk, but it's something that our viewers are really interested in, so I thought it was worth going back to. So does IgA run in families? Are people likely to pass it on to their children? That's what I want to know.
So it's a really good question. It's very clear that familial clustering of IgA nephropathy is well recognised. So if you look at a population of people, then there will be certain families that crop up in which multiple people in the same family have got the disease. Now, this has been well described in the medical literature going back at least 25 years and probably more than that. And these families have been very extensively investigated with genetic techniques which have moved on a lot over that period of time. And we haven't yet found a really clear causal gene. So it's not like eye colour. It's that family has a high background risk of IgA nephropathy, and that risk may manifest itself. But it doesn't mean, and with each generation, the risk, one presumes, will be getting less, provided the family is not in a geographical isolate, in which case it's a little bit more complicated. So yes, IgA nephropathy can occur in multiple family members, and it's well recognised, and it probably reflects families with just a slightly higher genetic risk of IgA nephropathy, but it's not really been proven to be a directly inherited trait like eye colour, where somebody whose parents got IgA nephropathy has, say, a 20 or 50% chance of the disease. That's never been shown.
Are you looking at the impact of epigenetics on IgA and looking at the influence of sort of the non-coding parts of the DNA and how they are controlling sort of gene expression? With the link between sort of what was the thing about the worms and things and sort of the environmental impacts on why more people in Southeast Asia have IgA compared to over here?
So that's really two questions, and I'm going to answer your question about the non-coding regions of DNA, and to tell you that almost all the genetic variants that have been associated with IgA nephropathy are in the non-coding regions of the DNA. And I think that's something that's come out of other similar studies in other diseases, that it's not the actual gene that's different in people with the high risk compared to the low risk. It's the amount of the gene that's made. And the part of the genome that controls or that has an important influence on how much of any particular gene is made is the non-coding regions near to that gene. And we are sort of at a very early stage scientifically in being able to understand how changes in the sequence of the non-coding parts of the genome affect gene expression or other aspects of gene regulation. So, for instance, the galactose-deficient IgA, the IgA glycosylation differences are very clearly coming from a non-coding region that's near to this one gene. And differences in that one region, and that's work that we're doing in my lab at the Royal Free at the moment, are very clearly have an effect on the amount of that protein that gene that's made by the cell. So yeah, non-coding sequence is absolutely critical to getting the balance of gene expression right in the cell. Epigenetics is much more, it's quite a different scientific field really, because again, that's a mechanism for control of gene expression, but it's not fixed in an individual for their whole life, and therefore it's not always clear that what's cause and what's effect. So it's a much more difficult problem to solve, and probably the stronger information that we've got at the moment comes from genetics, and our ability to understand epigenetics is quite rudimentary at the moment.
Okay, thank you. In the interest of time, I think we need to press on. Okay.